people from all over. So people are sharing in the chat where we're from, where we are right now. And I am really excited about this one. I have had Dr. Chris Beck on our list for a while, and I tell people, and I am not lying, that I really want to be like Chris. He does all the exciting stuff that the microbiome, cures, assessment. Here's a research coordinating network doing instructor training. It's such cool stuff. So a little bit about Chris. He's professor of pedagogy at Emory University in the Department of Biology. Is that correct?
Yep.
This is my old place in Rollins building, Rollins Research Center. And ever since I met Chris and heard about everything Chris and Chris's lab are doing, they are fantastic collaborations that include both the course-based research experience as well as the really thoughtful, both at the class level and at the faculty level assessment of course-based research experiences. And he has expanded and done so much. So there are course source articles on how to analyze microbiome data. There are tons of really cool things. And I am excited to learn what Dr. Beck is up to. And if you want to check out some of the many resources, this may be an older website, but some of the many resources the Beck Lab has made, check out a recent publication in Course Source that I will link at his website. So welcome, Chris.
Thanks, Carlos. If I had known he was going to link in my website, I would have updated it, but that's all right. Thank you all for being here on at least on the East Coast of Friday afternoon. Thanks for the invitation. The timing worked out perfectly. I'm currently missing our departmental faculty meeting. So, any excuse to miss the departmental faculty meeting, which for some reason we have on Friday afternoons. The only consolation is they normally offer us wine and cheese at our faculty meetings. I think that's the, only way they can convince us. So, just so I get a sense as to where everybody is in terms of teaching course-based research and sort of disciplines as well. Can I just have sort of a show of virtual hands? You can either like do that, or you can figure out the little reaction. How many people have taught a cure or are currently teaching a cure? Okay, so a decent number. Okay, good. And then maybe in the chat, because this is a little bit more difficult, what discipline are you in? Are you in biology or chemistry or physics or geosciences or something like that? All right, so a lot of bio, some chemists, got a physics and astronomy. All right, some neuroscience and so forth. All right, exercise science, good. All right, bioinformatics, A&P, okay. All right, so lots of bio, but some variety in there too. All right, good. So my goal for this session is actually not to tell you a lot about what I do. I'm happy to chat with folks about what I do, but more to help individuals and us as a group sort of think through finding the cure that works for you, or adapting existing cures or developing cures so that you can try it out and engage with this pedagogy in your own classrooms. I will say throughout this, if you have questions, feel free to interrupt me, unmute and interrupt me, throw a question in the chat, anything like that, right? So I do have some slides I'm going to share, and we'll start that way. There's a few things that I want to sort of talk about as a big group, and then I'm going to divide us into some breakout rooms to have discussions at a smaller group level, because there's a fairly big group today. So all right, so hopefully everybody can see my screen share. All right, awesome. So the first thing that we're going to do is what's called a waterfall. I don't know whether people have done this before or not, right? So this is I learned this from somebody. So this is a chat activity, but this is where you put in your answer, but you don't enter it right away, right? You type it in, but do not hit, don't send it, right? Because I don't want you to be biased by everybody else's responses. All right. So first, I want you to think about both your institutional and your course context. And by course context, I'm thinking about, what course might you teach a cure in? Is it an intro course? Is it an upper level course? Is it for majors? Is it for non-majors? What background would your students have? By institutional context, a lot of us are at very different institutions. Christy and I are across town. Christy's at Georgia State, which is a large state university. I'm at a private research university across town, right? So our institutional contexts are different. So I want you to think about your institutional and course context, right? And now type in the chat the most important component of a course-based undergraduate research experience for your institution and context. All right. So type that in, and then I'll tell you when you're allowed to hit enter. All right. Okay, you can hit enter now. All right, so I'm going to sort of scroll through these. So we've got some student buy-in, right? So yeah, we want students to buy into this research, engage with it. We think critical thinking, authentic research experience, and we'll talk some about, what does that mean, right? What does it mean for students to have an authentic research experience? It's a first research experience for many students, original research, right? And we can kind of think about what does it mean to do original research, some independent thought, right? Doing research that's new, right? And some folks are talking about some pretty big classes too, like 70 students or more. How do you do it, right? So experimental design, is it scalable? And some people thinking about it as sort of preparation. How do we use these experiences to prepare students for then to do independent mentored research, right? So, all right, and some scientific process skills, understanding research, right? Student agency, all right, and gateway courses. Okay. All right. So, cures, right, or course-based undergraduate research, we're trying to think about, what is it like to take a research experience that a student might have in a mentored research lab, working with a faculty member, and put that into a lab class, right? So that we can extend it beyond 1 or 2 students that, or maybe a handful that we have time and space to engage with in our research labs, and scale that. Some of you were talking about these gateway courses that can often be quite large, right? So, how do we do that? How do we manage that? And I think most importantly to start with is thinking about, what is in those courses, right? And what might differentiate those courses from non-course-based research courses, right? How do we differentiate it from like a standard lab class, or how do we differentiate it from, what people often call an inquiry-based lab class, right? How do we tease apart what these differences are? And so, some folks put up some ideas about, things like experimental design or original research questions, and that type of thing. So, I think this is a question that, the lab education community has been thinking about for a good while, and there are a variety of different thoughts as to what this actually means, right? So way back in 2014, it's hard to believe it's almost been a decade, right? With a colleague of mine at Emory and some colleagues elsewhere, University of Georgia and Radford University, we sent out a survey to a whole bunch of biology faculty, and we basically asked, what are the essential components of an authentic research experience? Just that open-ended question. And then, got back those responses and we coded them. And in the schematic here, you can see the types of things that people talked about, right? Things like, experimental design, students generating questions, collecting data, analyzing data, and also this idea of sort of novel research questions, right? Original research. But the really interesting thing to us that came out of this analysis is that respondents to this survey who talked about things like experimental design and data collection, all those things that are sort of grouped in that process of science box, never mentioned, or very rarely mentioned in their answers, anything about novel research questions or original research. And then there were folks whose only response was, it's got to be novel research, and didn't talk about the process of science things at all, right? So it seemed to us that, there are sort of 2 different approaches that faculty who are trying to consider authentic research in their lab classes, 2 approaches that they take, right? They can either emphasize the process of science part of it, or they emphasize the novel research part of it, but often don't engage with both of those, all right? Or at least when they're thinking about planning it, right, they're thinking about one thing or the other, but not both simultaneously. Doesn't mean that they don't do both, right? But they're focused on one versus the other. So at about the same time, a group called CureNet got together and had a big discussion about what is course-based undergraduate research, right? And sort of what defines it and how does it differ from other types of lab teaching. And so in this paper that they published from their meeting, right, they highlighted those 5 things that are listed on this slide, right? So using these scientific practices, the idea of discovery, it has to be broadly relevant, that students have a chance to do iteration, which basically means if it didn't work the first time, they get to do it again, right? And then the idea of collaboration. So they sort of put forth these 5 things as, markers of what a course-based undergraduate research experience would be. a little while later, another group, in this case, it was a collaboration of institutions that had funding from HHMI to institute undergraduate research in courses. And in this case, it was beyond biology, it was across disciplines, right? So it was physics and bio and chemistry and math and engineering and so forth, right? And from a broad range of different types of institutions. We're sort of thinking about, the context that had come out and, how that fit with their institutional and course context and what they do. And they came up with this schematic here, right? This idea of sort of a wagon wheel, and you can see some of the terminology that shows up there, right? That was on the previous slide. So things like, iteration and discovery, but some of those science process things that showed up in our earlier survey. The other thing is, as they were envisioning this, is that they put it on a wheel for a purpose, right? And their idea was we could take out one of these spokes, right? And the wheel would probably still work, right? That we don't necessarily have to have all 6 of these things to be able to, call it a course-based research experience, and for students to have positive outcomes as a result of it. So we could take out a few of these things, right? I think we always struggle with this, whether it's a lecture class or a lab class. We can't do everything. So how do we prioritize? And so, we can sort of think about that prioritization. But, they also consider the fact that if you take out too many of these things, the wheel doesn't work anymore, right? So it's not like you can, just focus on one of these things or 2 of these things, right? That you need many of these things, but we're not quite sure, which ones are the most important. And in different institutional contexts, in different course contexts, you might want to emphasize some things versus others. Whether it's an intro class, right, or an upper level class. So, as Brenda points out, ownership is super important, right? And so, yeah, I think there's good evidence that student ownership of the process is important. I would say we're still figuring out what's the best way to create that ownership for students, or whether there are multiple ways that students, feel some sense of ownership over what they're doing, and how that connects to sort of other of these elements. And then this was more recently, I would say, maybe 5 or so years ago. Another research coordination network started thinking about competencies in biological experimentation, and came up with this lollipop model, right? And so, again, this is more thinking about what students need to do to be competent at experimentation. It is focused on biology, so some of these things might not make sense in the context of physics, or might not make sense in the context of chemistry or something like that, but they might make sense at this broad level. Each of these lollipops has sort of sub-elements to it, which are more biology specific. But this was another way of sort of thinking about how we can engage students with undergraduate research in a class, is thinking about these different competencies in biological experimentation and how we go about implementing them in a class. And so it's a different framework for thinking about this. And then there's some other ideas, right? So there was a paper that came out of Aaron Shortlich's group at Portland State that I found really interesting, that students think that their research-based course is authentic if they fail. If everything works perfectly and they don't fail, their students' perception is, I'm not doing research. And that, of all of these things that they looked at, the most important thing in affecting students' perception of I'm doing research is that something didn't work, right? That they failed. Which I found that really a really interesting result, right? And in those original definitions, we don't think about that. It's sort of baked into the ideas of iteration, right? You have to iterate because something failed. But that idea of iteration. And then that continuum down at the bottom of this slide comes out of the chemistry education literature, and sort of how at least some folks were thinking about how what research looked like in chemistry labs. And I think the interesting thing here is that there's this continuum of student responsibility, right? Going from less student responsibility, sort of these traditional cookbook, what they think about as traditional or cookbook labs, right? To, much more student responsibility. And, research-based classes is more to that extreme of more student responsibility, right? And thinking about that continuum, we'd have to think about, what does student responsibility mean, right? Or, good question, Aaron, right? Is that, autonomy, are they deciding that research question? Or, what aspects of it are they responsible for, right? Aaron, I didn't actually, pay Aaron, but I think my next slide sort of goes into this. Yes. But thanks, Aaron. That was great. So this is sort of another framework that overlays that autonomy or that responsibility framework, right? Over top of cures, right? And how we can kind of think about that student autonomy, right? And so this schematic and framework, that I've been thinking about for years with my colleague at Morehouse College, Larry Bloomer, that there is this continuum, and at sort of the bottom of this matrix is students have very little autonomy, right? That the faculty members defining the research question, right? The experimental design is defined by the faculty member. And this might be driven by faculty research interest, right? I'm interested in this thing, so your students in my class are going to do the thing that I'm going to tell them to do, and this is the experiment they're going to do, and they're going to collect these data for me, right? But we might still think about it as a research experience, because maybe they're addressing novel research questions. We don't know the answer to them. And in many ways, this is how mentored research in a lab often works, especially for beginning students, right? That at least in some institutional contexts, when an undergraduate student walks into our lab, we say, all right, there's this research question that our research group is working on, and we want you to work on this part of it, right? And we're going to show you the methods for doing this, and we're going to, tell you the experimental design, and we're going to train you to do this, and you're going to collect these data, right? We can give students a little bit more autonomy. We can, say this is the research question, but the students actually have to design the experiment, right? So, as we move towards the top of this chart, students get sort of more and more autonomy. I've also sort of embedded in there this idea of community, right? So sort of community engaged learning. And so we can go beyond the bounds of our institution. And I know many people do this. And I think that like this Friday Falcon talk from 2 weeks ago, wasn't there discussion about like sensor and the sensor network and, community engaged learning and things like that, right? So that fits into this framework very nicely as well, right? And then, way up at the top there as sort of the most, the most student autonomy is that students get to decide on their research question. They get to design the experiments, right? And we bound it in some way, right? We, has to be within the discipline, or we select some research papers and say, all right, read these first and now come up with your research question, and now design your experiment, and now you're going to do this thing, right? So Brenda wants to know, would you put a culminating project that applies techniques learned in the class at the top of the list? It's bounded by a variable. Yeah. So bounded by materials available. I would say yes to that. So I'm an ecologist by training. I teach an upper level ecology lab class here at Emory, and during the first half of the semester, we do things more at the bottom of this list, where I give them the research question, and the students design the experiment, and then we go out and collect the data. And I would say one of the nice things about ecology is, even if we do the same project across multiple years, the data will always be different, right? Because it's nature, right? And we go outside to the woods, and the weather matter, like everything matters, right? So we don't necessarily always get the same answer. But the second half of the semester, students work in groups to design their own projects, come up with their research questions and their own experimental designs. And, the only limits I give them is, here are the materials you have available. It has to be ecology, and you have 6 weeks, figure it out, right? All of that said, this runs this continuum. And so, some cures, and we'll talk about some examples in a minute, fall way down at the bottom of this, right? So where that research question is defined for the students, the methods are defined for the students, right? The students actually have very little autonomy over anything, or essentially no autonomy over anything, but they're still doing research, right? And I would say, the impact of that degree of student autonomy on student outcomes in cures, I think is sort of an open question, and something we're trying to figure out. We do know from research that's been done on cures, where students basically have no autonomy, students benefit, right? They gain from that experience, even though they have very little autonomy. Whether they would gain more with more autonomy, we don't really know. As Carlos mentioned, the cure that I'm working with now is on insect microbiomes, and the way that we're working with faculty is that after we've trained faculty on this cure, they are implementing it twice for us. Once where the faculty member decides on the research question, and a second semester where the students define the research question. But all the methods are the same, right? And so we're hoping to get at, how much does that autonomy in terms of defining the research question in the context of this, research model affect student outcomes? So when, I think when faculty are thinking about cures, it's important to keep in mind these different approaches to cures, and it doesn't have to be that top end, right? It can be that top end, but if you don't feel like it fits your course or your student population to give them free rein, that's fine, right? It could be at the bottom of this, and we would still consider it a course-based research experience. Okay. All right. So what are some different models out there, right? So there are, at least in biology, there are a bunch of what we call national models, right? So, perhaps one of the, best known one is the C-phages project, right? That HHMI has funded for years. And, if I go back a slide, C-phages is at the bottom of this table, right? Students have essentially no choice whatsoever about what they do, right? They go out, they collect soil, they isolate phage from it, right? They sequence that phage and they do a bunch of bioinformatics, right? Students might get to choose where they get the soil from, right? But, beyond that, there's essentially very little student autonomy. Right. There's the genomics education partnership, the bean beetle microbiome projects, that I work with, things like Tiny Earth and the PAIR project, right, are these sort of national models. And so, for some faculty, one easy entryway into a cure is to join one of these national models, right? Folks have worked out how to implement this in your classroom, and provide the resources and the faculty professional development for doing that, right? Some of these are longer. C-phages is set up as a 2-semester thing. The genomics education partnership can be done in different durations. The PAIR project's actually only a couple weeks, right? So you can sort of fit it in, right? And then I think, the other approach is using either institutional or individual research projects, right? So generating internally from your faculty, either yourself, hey, I've got this really cool idea. I need a team of 24 students to help me collect these data. Let's do this thing, right? Or what we do in our introductory biology labs here at Emory, where we have about 600 students a semester, is that the research projects that students work on come out of the research labs of our research faculty. And so students do short, like between 4 and 6 week research modules that are based around faculty research. And again, this could also be community context too, right? You've got some community partner that helps you sort of define those, define those research projects. So there's a bunch of different ways to do this. All right. All right. I'm going the wrong way. All right. So before we go on to this, right? Does anybody have any questions? I'm gonna like be quiet for a minute, and Aaron and I are gonna chat later about microbiome ecology. That's awesome. Anybody else have any questions? Okay, so question for later. Anything that smells like a lab cure is immediately labeled as expensive and prohibits development of courses to have wet lab included. How have others successful in finding money for cures? Right. Yeah, we can definitely definitely talk about sort of funding. One of the things, and this is the bottom bullet point there, you know, what are some of those barriers that you need to overcome? And one is this, you know, mindset that these things are expensive, right? And two is, where can you, where can you find funding, to fund these particular projects, right? Resources for a genetics lab that meets 4 hours a week. And so, Katie, are you thinking about, something? I suppose I should have had a slide on this, right? Some people, I don't know whether I would call it a common misconception, but the thought, some people think, oh, this has to last the entire semester, right? And students have to be working on the same project for the entire semester. And in some cases, that's the way that these cures are implemented. As I mentioned in our intro class, we do different ones for, 4 or 5 weeks at a time. In a lot of, I know a lot of cases where folks spend the first, half of the semester doing more traditional labs where students are learning techniques, and then do cures sort of later on in the semester, right? And so, Katie, maybe you can help us a little, and maybe there's some geneticists in the virtual room here who have some ideas of, what are you thinking about? Are you thinking about something for the entire year or an entire semester or part of a semester?
Right. Thank you. I appreciate it. So I teach a genetics lab that's writing intensive, and we meet 4 hours a week, which is kind of difficult because it's usually a 4-hour chunk. We meet 16 hours or 16 weeks in our semester, but we don't need to have it running through the entire semester. We just want to give experience, right? It's kind of challenging, at least for me, because I can't wrap my head around doing a cure in such a small amount of time. But if there are smaller things out there, I am all ears. So thank you.
Yeah. So, Robert said that there's a Sabre undergraduate genetics cure group, right? That might have some, some good ideas. And I think it depends on what type of genetics you want them to be. Do you want them to be doing molecular genetics? Do you want them to be doing, more bioinformatics type things? More molecular biology, or that type of, those types of things. Are you completely open?
Well, I'm open to a certain extent, but I do know that if we do bioinformatics, it doesn't always get well received by the students because we meet so infrequently that they're coming in with a sophomore level knowledge, and I don't know, like maybe we don't have the right bioinformatics. I don't know.
Yeah.
They like getting their hands wet. They like the pipettes. They like the molecular. They like watching and observing. It's more touching, I guess, versus
Right.
the computer. At least at their stage, our population feels more closer when they can actually manipulate tactile items.
No,
Not saying you can't do that on a screen, but I just
right. No, I agree. For some, at least for some students, the bioinformatics or the computational is like this black box, and they're just pushing a bunch of buttons, and they don't, even though clearly that's research, right? They don't necessarily see it as such. They see it as, they see it as, as something different, right? And that for it to be researched, they have to be pipetting things, right? And mixing reagents and, that type of thing. We'll work on digging up some some possibilities. Some of these these national projects involve aspects of sort of molecular biology and molecular genetics, right? Whether that's basic PCR and sequencing, or more next gen sequencing and that type of thing. The genomics education the genomics education partnership is more computer based, right? Because that's genome annotation. So all right, but we'll some folks have put some stuff in the chat, to some of which are related to genetics. So take a look there, and we can see if we can crowdsource some other ideas. All right. All right. So let me just breeze through these chats. So a bunch of good ideas. Everybody, some people said, call Carlos and he will give you lots of money. I think that's what that said. Or partner with Carlos, right? Right, or partner with each other, right? Yeah, and Lawrence has talked about done genetics-oriented cures with Arabidopsis, Drosophila, and then C. elegans, and characterizing mutants. So there's some possibilities there. And Usha wants to know how you measure student learning gains. And that's at the end. So we better go on to the next part so that they're, oh, wow, where does the time go? So what I want you to do, I'm going to break you up into breakout rooms, and we're just going to do this for a very short period of time, but so that everybody has a chance to talk, right? So there are those questions there that I want you to think about. I'm gonna, what I discovered recently is that when I put people in breakout rooms, that the chat disappears when you go to the breakout rooms. And so I put these same questions on a slide. All right. And so there they are. So I'm gonna give you a minute to find that, and I'm gonna look. All right, we have 36 people. So I'm gonna make some breakout rooms, and randomly assign people, just take a few minutes to talk amongst yourself about some of these ideas, and then we'll come back. I can stay a little bit longer, and we can get to some of these other questions as well. So, all right. So I created a bunch of rooms. I'm going to open them up now. So hop in, spend a few minutes chatting about those questions, and then we'll come back together.
Are you gonna go hop some rooms?
So I can hop into a room. All right, so I know I gave you just enough time to just introduce yourselves, and you maybe half of you got through question number one. I was mentioning to Talitha, that when I typically do this, I do it across 2 days, right? Like 2 full days or a day and a half or something like that. So my hope is that in this session, you've gotten some ideas, right? To start thinking about it. You know where I am now. And so you can send me emails and, ask me questions. I do want to end with Usha's question. So Usha was interested in outcomes, right? I'm gonna share my slides again. Here are some options, right? We do not do all of these, but these are a variety of things that you could do. It depends on what you're interested in and sort of how you want to go about doing it, right? If I can figure out how to do it here, I'm gonna actually drop a couple of files in the chat that relate to assessing student outcomes. So one is a list of various types of student outcomes, such as this, along with references to papers that have instruments that you can use to assess these various outcomes. And then the other is a book chapter that I co-authored with my colleague Larry Bloomer at Morehouse and our postdoc, who is now a faculty member at Cal State San Bernardino, where we looked at these various instruments for assessing cures in the context of that biological experimentation framework, and what aspects of that framework do they cover versus don't cover. Let me see if I can throw these in the chat, because one is a document I put together, and then the other is, since it's a book chapter, it's a little bit harder to find. So there's the book chapter. Here's the book chapter. Right.
Chris, are you okay with us posting these in the Slack under your?
Yep. Yeah,
meeting?
that's totally fine. The book chapter is actually open access. It's just because it's a book chapter, it's a little bit hard to find. And then this other PDF is this list of cure assessment instruments. All right. Just so you can have some resources, again, feel free to reach out to me. I know that some folks probably need to go because we're on the hour here, but I'm happy to hang out for a little bit and happy to answer any questions that folks have. Christy is suggesting it'd be great to have a list of who is doing what kind of cure. If people would share, could we start a Google spreadsheet or something to put the info in, course level, focus, etc. That's a good idea. I don't know whether Carlos, you want to take that on. Right.
Yeah, I'm thinking that's a great idea, and I'm trying to think if there's.
Yeah, there's is the CureNet list, right? Which is a list of a bunch of cures. I'm not sure whether that list actually has who is doing those cures, right?
Yep.
So
I could share a Google Doc and we can start.
Yeah. Hey, John, what's up?
Thank you so much, Chris. This was a really helpful presentation, and I'm really excited to also hear all the different examples that other faculty had in this room. One question I have is, so I'm supporting faculty who are trying to develop cures, and one question I had was for faculty members who are currently teaching very traditional lab courses, where students are pretty much following the lab manual. Would you recommend that they first start off with more inquiry-based labs before moving to a cure, or do you think it's okay for them to jump straight into a cure? I was just wondering what the learning curve is in terms of developing one and implementing one in the classroom, and whether there are
Right.
ways to kind of scaffold that process.
I think it depends, I guess my view is it depends a little bit on the comfort zone of the faculty member themselves, right? And so some are, going to be more comfortable with that change than others. And so, for some, it might take those baby steps, right? Of, all right, this is, we've opened it up a little, this is more inquiry, whereas others, could essentially dive right in, right? I do think it is important to, I think, faculty who've not been involved with cures think it's the extreme part of that continuum of student autonomy that I showed, right? That students are, wide open, given the question, they can come up with anything and do anything and so forth, right? And I think to many faculty that's scary, right? Because it's like, oh my gosh, I'm going to have, this room of 24 students, even if they're working in groups of 4, we've got 6 different projects going on and they're all over the map and I've got to manage all of this, right? To let them know that not all cures are the same, right? That some of them are, very low student autonomy, right? That, but it's not cookbook in the sense of everybody knows what the answer is, right? This is not, we did mitosis in intro biolab, so now we're going to look at onion root tip and look at cells that are undergoing mitosis, right? It's not that, but it doesn't have to be, like, what do you want to do, right? The other thing that I've found that faculty have to come to grips with, and is that the learning outcomes of lab are different than the learning outcomes of the lecture class, and that the role of lab is not to recapitulate what was just talked about in lecture, right? That the old model, right, is we do this in lecture, then we do sort of a confirmatory lab on the exact same thing, that week or maybe the next week. And it's this one-to-one correspondence as we go throughout the semester, right? Cures don't fit into that model, right? And, they won't. That we have to, articulate for ourselves and for our students that the learning outcomes of lab are very different, right? And that the goal of this lab is not to, look at mitosis, right? I showed you pretty pictures on my PowerPoint slide. You don't need to look under a microscope at that, right? What helps even more is classes in which the lab is a separate course from lecture, right? And so so rather than the traditional 4-credit course, it's a 3-credit lecture and a 1-credit lab. They get separate grades, right? And so, it's easier to reinforce for both faculty and students that, this is a different endeavor, right? And that the outcomes, that we're interested in are different. And so it's okay if we spend the entire semester, working on this research project that, is maybe related to genetics, but, we're not spending the entire semester talking about genetics or something like that. So, So those are some ideas.
Thank you. That's really helpful. Thanks
Yeah.
for your insights.
Yeah. Others? All right, Carlos started the Google Doc.
Uh-oh. Uh-oh.
And so presumably, Carlos, there's a way to share this this Google Doc with everybody who signed up for this, or the broader community or something like that, so people could add on if they wanted.
Yeah, we shared it on the Slack, but that's a good question. A question I'll ask Melanie and Talitha at some point.
Okay. All right.
Melanie, will you send it to the Twitterverse?
You got it. I'm on it.
So, there was the question earlier about funding cures, right? And, and so some of it is collaboration, right? But I think, even traditional lab classes use resources, right? There's no free lab, right? Maybe if it's purely computational, but you could use those same resources, but the questions that students are asking are different, right? The questions are ones that are not routine and anybody could look up on Google, or if we ask Chat GPT, it might get it ready right. The Google one wouldn't get it right. We figured that out the other day, that the Google bot screws up answers, but at least in astronomy it screws up answers. So I think it's being creative about how do we use those resources that we have, the equipment that we have, that we would traditionally use, but use it in in a different context. And and I would say also, don't go into, I would say, don't go into it with the expectation that your students are gonna, necessarily generate publication worthy data, in 4 weeks in the semester or something like that, right? We all have worked in research labs over the years at some point in time. We know, that type of science can't be done in 3-hour lab periods and so forth, right? So what? So Carlos, is this question for me?
That's for you, and it's something that I've had on my mind for a little bit. I'm like, this workbook is really cool. Or
What? So what workbook are you talking about, Carlos?
the PDF. I guess it's almost like a lab manual.
Oh, like the Bean Beetle handbook?
Yeah, the handbook. That's the word.
Yeah. That was really, we developed that for faculty who wanted to use bean beetles in their teaching and needed to know basic natural history and basic, sort of lab techniques, right? There aren't any labs there, right? It's just, what are these beetles? What's their natural history? What, how do you rear them in the lab? How do you tell males from females? All of this type of stuff, right? And so we give that to faculty, and they, in our workshops, that relate to bean beetles, so that they have that as a resource.
Yeah, and Aaron, the metadata, the handbook, that made me think about this spreadsheet. Eventually, if some people may not write the handbook right now, but to have that in the back of your head or someplace here, that it's a really great way of getting others, I won't say up and running, but really interested in, I can do this. Here's some of the background, why it's interesting. I can take on
Right.
beetle microbiomes.
Yeah.
What questions do you have? And I know we're over. And well, thank you, Chris. You gave us lots of ideas.
Well, thank you all for coming. And as I said, if you have more questions, feel free to send me an email. I'm happy to email chat or Zoom chat with people.
I'm curious how you disseminate things. Like, do you have specific journals that you target? Or my first thought is, well, if I put it on Open Educational Resource Commons, then it gets a DOI, and then it's just about like, here's this link I can blast into the world.
So, I think there's different avenues for like the curriculum itself, right? Versus, assessments of that curriculum or of student outcomes, students involved in that, right? And so, a lot of the curriculum stuff, I disseminate through the Association for Biology Laboratory Education. That goes by the acronym ABLE. And it's associated, the publication is associated with the annual meeting. And I love the annual meeting because it is a workshop meeting. So you go, if you're presenting, you go and tell everybody about your lab, right? And they actually do it. They do the lab during the meeting, or some part of it. And then later, you can publish that, right, in the proceedings. And instructors' notes are there, and student handouts are there, and all of that, right? But I like it because you can, you're actually having folks work on it, and you have this back and forth of, oh, well, you teach it at Emory, I teach it at 2-year college. How would you, do this? Or I need to scale this to 1,000 students. How, because I teach at NC State, right? How do I do that? Right. And that type of thing. I think CoreSource is a good one. As an ecologist, we have teaching issues and experiments in ecology, or TIE. I'm the editor of that. And so there's the experiments part, right, which is where cures or lab and field-based stuff could go. A lot of the education journals, like Journal of Molecular Biology and Biology Education, so JMBE, and the Biochemistry and Molecular Biology Education, publish just curriculum. You don't have to have any student assessment data. You can, they have sections where you just publish the curriculum. American Biology Teacher does as well, right? There are similar things in other disciplines. And then, if it's student outcome data, I think there's a variety of, places. I tend to have published in CBE Life Sciences and also JMBE. A lot of folks also publish in PLOS One as a place for sort of student assessment data. American Biology Teachers publishes some of that. Let's see, Journal of Research and Science Teaching also publishes some of that type of stuff. And then there's sort of scholarship of teaching and learning journals sort of more broadly that, you know, publish that type of work. So, and I think it also depends on what position you're in, you know, and how important research publications are and what counts, right? And that, like, just putting it as an open educational research source and getting a DOI, you know, in many contexts, you get zero credit for that, right? People are like, yeah, you know, it's not peer reviewed, you know, I don't, you know, yeah, you have a DOI, but I don't care, right? Whereas, you know, if it goes through peer review at core source or, you know, some of these other avenues, then, you know, it's looked upon more favorably, right? But if your position is such that, you know, nobody cares about that, then, you know, can do other things.
Good question. Any other questions?
All right.